topic which I gave for this program is kind of multiple topic, particularly two different diverse topics. One is about origin of our own. And I think it's very curious to all of us that know that how we all have originated. The second one is about our health and disease. So I'm going to combine both our origin and health and diseases. So evolution and health and diseases. And if you want to know why I brought this together, and you'll be happy to know that Debzhansky is a well-known geneticist from Rockefeller University. He wrote an article in the American biology teacher, and the title is "Nothing in Biology Makes Sense Except in the Light of Evolution." I think towards my talk, you will understand how important this statement is. So let's go back and see how Darwin envisaged about 160 years back. He wrote a book called The Origin of Species in the year 1858. And where he has described about evolution of most of the organisms, right from archy to whales. But what was missing in that book was that how the human might have originated. 5 years later, Thomas Henry Huxley, in the year 1863, he wrote a book. I think that's the very first book talked about origin of human. Say that there was found evidence about evolution of human, that human and ape are evolved from a common ancestor. 8 years later, Darwin wrote another book called The Descent of Man, where he has declared that chimpanzee and gorilla are our close relatives based on the anatomical similarity. But of course, now after the human genome and many other organism genome have been sequenced, it's very clear because the human and chimpanzee share about 98.8% of our genome, and it's very evident that the human and chimpanzee have evolved from a common ancestor called hominids. Okay? But most of us accept this, but there are some resistance. Even some other people who are in very high position, responsible people, they say that nobody has seen that chimpanzee turning into human. Because it's not a magic that chimpanzee will turn immediately into human, because it's evolutionary is a long process. It takes millions of years. So if you see what happened during the last 7 million years, actually this is the period where human and chimpanzee have diverged. There are many species have evolved and they all disappeared. But I just focus this into very closer, divided into two parts. In the process of human evolution, last 7 million years, as I mentioned, there are several species have evolved and they are all vanished. And this process, there are three major differences we acquired. One is walking with 2 legs, called as bipedalism. We have ability to walk with 2 legs, and also ability to make tools for hunting animal, search for food and so on. The third important aspect is that our brain size has increased. And if you look at the early species, chimpanzees, the brain size was as similar as chimpanzee. Then it has increased to 35% in Australopithecus. In Homo habilis, 50%, Homo ergaster, 60%. And if you look at the Neanderthals, our very close relative in terms of evolution, the brain size was predicted as slightly larger than human. But nobody knows whether they are intelligent than human. But interestingly, one of the evolutionary biologists, Paul Mullis from Cambridge, he wrote in one of his articles, the reason for why, one of the reasons for why Neanderthal is no more, is vanished, he says that inherent violent behavior of modern human probably responsible for vanishing of the species. Could be true, right? So, this is how we have evolved. And as a geneticist, I always believe that the genetics play a major role in everything, and of course directly for the evolution. The DNA change which brought different phenotypes, right? For example, this is a human Y chromosome, just taking only one of the chromosomes to show that what are the changes happened in terms of some of the genes. In chimpanzee, bonobo, gorilla, and many other species. And if you look at the gene called SRY, that's a sex determining region on the Y chromosome, that's essential to make individual as a male. If there is a single base mutation, that will turn into female phenotype. That gene is present in the P arm, the short arm of human Y chromosome, whereas in chimpanzee is the long arm. Okay. And again you can see in gorilla P arm, as you see this highlighted, they are in different position. And this is one more example of a few more genes like chromodomain Y, deleted azoospermia, they are all in the long arm, whereas in the short arm of chimpanzee and bonobo. As you see here, there are a few copies moved to short arm also. So these are the kind of changes happened during last 7 million years, which brought the phenotype the way we look like today. And even if you don't think that did not happen, but you look at the 3 brothers who are born to a normal parents, okay? And what happened? Probably whatever the changes we acquired to become like a modern human, the way we look like today, and there was a back mutation which brought something like this. There are a few more examples. You can see this gentleman, and you can see this child, and there are some features like the body full of hair, which we have lost during the course of evolution, is brought back because of the DNA changes. Then the question is that how the unique features evolved, and how did the capacity for language, reasoning, culture encoded in the gene evolve? Because these are some of the unique features like reasoning, language of human. As I mentioned, the genetics played a major role. A mutation in a gene called myosin heavy chain, which created more space in the skull, therefore the brain size has increased. And there are few more genes. There's another gene called protonorephin, which involved in learning, experience of pain, perception, behavior, and memory, has been identified. FOXP2 is another gene which is essential for normal human speech and language. Gene must be the same, but the mutation in which brought these kind of changes. As I mentioned, DNA play a major role in everything, including to decide the drug which we take that should be metabolized and give therapeutic response, no response, or adverse response. All of these are based on the DNA changes. Another gene called amylase that encodes salivary amylase, that gene has become multiple copy when the agriculture started during the Neolithic period around 10 to 11,000 years back, because to digest starch-rich food. Similarly, LCT is the lactate persistent gene. After we domesticated the animal, started drinking the milk, that gene mutation evolved even to help us in digesting the milk sugar even when we become adult. Otherwise, the enzyme work only during the weaning period. That's a kind of genetic perspective. If you look at the social scientists or anthropologists, what they think about human evolution, they said that human learn how to self-domesticate ourselves. You know that from the wild, we bring the animal, including dog or horse, and domesticate, they'll become very milder. But the human self-domesticated ourselves to become the cooperative species. So with this background, what I'm going to tell you is that how the modern humans have evolved. And it's very well-known fact that the modern human evolved in Africa about 160,000 years back. And even today, there are several scientific evidences. This very recent paper, 2019, found that the modern human arose in Africa. And from Africa, they have migrated to many parts of the world and populated. That why did they migrate? That's one of the questions was raised, but they are not the biologists, they are geologists who found that the water level has gone down less than 90% during the year 135 to between 135 and 75,000 years back. Therefore, no water for people to survive. Then they started exploring and started migrating out of Africa. So how did they migrate? There are two hypotheses. Archaeologists, anthropologists, historian, geneticist, linguist. One group of them believed that the first modern human migrated out of Africa taking the northern land route, because there's a land connection and they must have migrated. But other group, they did not believe on that, because the climatic condition in Europe, even today it's very difficult for people to migrate. So they believed that the first modern human might have migrated taking the southern coastal route. And these are all based on the archaeological evidence present in some of these countries or continent. Evidence in Australia suggests that the modern human must have arrived about 50,000 years back. In China, 39,000 years back. In Middle East, 47,000 years back, and Europe, 40,000 years back. But unfortunately, we don't have any good archaeological site to get some biological material to understand when did the modern human arrived in Indian subcontinent. There are some excavation that was in Jolapuram, Andhra Pradesh, that was published in 2007, that they excavated and found only the stone tools which probably the modern human might have used, but they did not find any biological material to analyze and confirm that. There are a few more sites in South India, in Tamil Nadu, near Madurai, place called Kiradi. There's a lot of excavations being taken, and 2 intact skeleton was also obtained. We are working on it. And there's a place called Adichanallur, again in South Tamil Nadu. So it's not going to give much information because the skeleton itself is not very old. One successful study, which again in collaboration with archaeologists in a place called Rakigiri in Harappan site, there was an intact skeleton again was obtained, and we sequenced the whole genome. And this is again is not very old. This is about 3000 to 4000 BCE as you look at it. And this is this Rakigiri sample, which consisting of 2 distinct genome. So one is the Indian genome, which I will come a bit later, what is the Andamanese one. The second one is Iranian hunter-gatherer, but not the Iranian farmer. There is one view that the Iranian farmer must have brought the agriculture into India, but in this case, probably that must have happened later, but there is no Iranian farmer DNA in that skeleton. So this is only Iranian hunter-gatherer, and sample is not very old, 3000 BCE. This is another very extensive study of more than 500 bone samples which are obtained from South and Central Asia. This study again is not giving enough evidence based on this study. This study suggests that there was a migration of people, Yamana pastoralites, and this contributed very limited to India, and that's again not very old, 2000 to 1000 BCE, and they also gone to Europe. Probably they are the one brought Indo-European language to both India and also to Europe. So all of these are not giving any clue about when did the modern human arrived in Indian subcontinent. In the absence of archaeological evidence, where you have seen 50,000 years back in Australia, 47,000 years back in Middle East, and so on, we thought that because we have very well diverse human population in India, and we thought, why not we analyze the genome and try to understand when did the modern human arrived in Indian subcontinent. We have been doing this for nearly 27, 28 years, but our surprise is that the first out of Africa human is in Indian subcontinent. They are none other than the tribal populations of Andaman and Nicobar Islands. I don't know how many of you are aware that there is a population like this in Indian subcontinent. To give you some idea about that, there are 572 small islands. There are 2 groups of populations in these islands. One, they typically look like Africans, that's the Java, Great Andamanese, Sentinelese, and Onge. There are 2 populations, Nicobaris and Champagne, that typically look like Southeast Asian group, like Chinese and so on. While we could be able to analyze Java, Great Andamanese, and Onge, Sentinelese, nobody can go near them, because once you go there, they will shoot with arrow, and that's what happened a few years back, that one American journalist tried to go there, but they have killed, maybe 4 or 5 years back. So to give you the idea about the population size, they are very small in number. Onge only 98, Java 200, Sentinel 250, Great Andamanese are only 20 or even lesser than that now. So one of the questions is that why their number is so low. One could be because they are highly inbred population, but other one is very surprising that when the Britishers captured India and wanted to construct the cellular jail, I don't know how many have visited the Port Blair to see the cellular jail. There is an entrance, and there used to be 7 wings, and now I think only 3 or 4 are existing. So this is the way they have constructed to put our freedom fighters. and somebody is there in one of the cell, they cannot see the outside the world. They can only see the front wall, and back side again, they're all hidden, and even if they can see only front side of this. So that's the way they have organized. So why I am brought this here is that when they wanted to construct the cellular jail, they killed most of the tribal groups. And I don't know what they thought at that time. After killing, they took their skull and has been kept in Duckworth Laboratory Museum. So we had a collaboration with Cambridge University, and we had access to collect some of the bone sample. This is one source of samples which we analyzed. The second one, we ourselves visited to, there's a place called Dugong Creek. That was long ago. And we collected the sample, myself, the technologist, and a few more of my colleagues. One has to go to Port Blair, from there to another place, Dugong Creek by overnight ship, from there by road, then a small boat, actually go like take a U-turn, because that's a secured place where they live. We collected the samples, and we used the genomic approach to understand about their origin and history. We used two different kind of genome. In a given cell, there is a nucleus where we have 46 chromosomes. We inherit 23 from father, 23 from the mother in the form of sperm and egg. And you also know that this power of the cell, they are mitochondria, and there is a large number of mitochondria in a given cell, and each mitochondria will have a large number of mitochondrial DNA. And the DNA of the mitochondria itself, we all inherit only from the mother. If you look at the inheritance pattern, just to give you the idea, this is a male, as a female karyotype, this is a male karyotype, there's a X and Y, there are two X's. And up to here, 22 pairs or 44 chromosomes are common between male and female, there's no difference. The only difference is the sex chromosomes. In female, 2 X chromosome, and male, 1 X and Y chromosome. This is what makes the individual as a male. In the next generation, this is in the somatic cell, in the body cell. In the germ cells, we have only half of these numbers, one of this pair and one of this pair. So instead of 46, there will be 23, which we inherit from mother and from father. There are 2 N, there is only 1 N here, half of it. But the way which is inherited going to every cell division is not fixed, either this chromosome or this chromosome, any one of these 2 chromosomes. So there is a shuffling. That is the reason that even there are 8 or 10 children in the family, everybody looks different, because the inheritance of these chromosomes are different, except in case of monozygotic twins, because they are exactly the same chromosomes divides. And if you look at the mitochondria, in the egg, there are a large number of mitochondria. In the sperm, the mitochondria is in the mid piece of the sperm. It remains outside at the time of fertilization. So therefore, every individual receives only the nuclear genome from the father, all the mitochondria from the mother. So that's one of the reasons why we use the mitochondrial DNA to trace the maternal lineage. So we sequence the mitochondrial DNA, and when you sequence DNAs, those who are not familiar with the DNA, there are 4 nucleotides, adenine, guanine, thymine, and cytosine, and the way it is organized, that's what makes sense. Like words is formed by different letters, 26 alphabet. I'm just showing that how we get the difference. For example, these are 2 mitochondrial DNA sequence, and mostly they're all common. Wherever there's a dot, usually there's a G, guanine, and here there's adenine. There's adenine here, there's a guanine here. Similarly, adenine and guanine. So these are change. Some of the change happens very frequently. Some of the changes are highly conserved. Okay, goes across different generations. So we pick up those variations and we make haplogroup, put set of variation that makes one haplogroup. I will not go into detail to confuse you, but what I wanted to highlight is that if you look at the pie diagram, that indicates that L1, L2, L3, these are the haplogroup which is exclusively present only in Africa, because these are all defined by one set of mutations. Okay. As we acquire mutation, in addition to that, then we fall into different haplogroups. Okay. That's how, if you look at either country or continent, or either one particular haplogroup or combination of haplogroup which is exclusively present there. In India, if you look at, majority of them are with M haplogroup, and if you look at Europe, or if you look at North and South America, and if you look at Australia, PNG Island, everything is very unique, right? So using the mitochondrial DNA sequence, we sequence the complete mitochondrial DNA and wanted to assign haplogroup. We expect that they would fall in L1, L2 or L3 haplogroup because they are typically look like Africans. To our surprise, they did not have any of those, they have the mutation but did not fall into that particular haplogroup. Rather, they all have unique set of mutation. Mitochondrial DNA is 16,500 base pair, and if you start from number 1, at the 200th position there is a mutation, 1524 there is a mutation. These are all unique mutation which was not found anywhere in the world. Okay. Therefore, we assigned a new haplogroup called M31 and M32, and one can also calculate the age of the population by using this information. And putting all of this together, for the first time, we concluded that the Andamanis are the first modern human must have migrated out of Africa, taking southern coastal route and reached Andaman and Nicobar Islands about 65,000 years back. This is the very first genetic evidence suggesting that the Andamanis are the first modern human must have migrated out of Africa. There was a lot of supporting evidence. In the same issue, there was another study, studied about the population called Orang Asli from Malaysia. So they also has come to that same conclusion. And in the same issue, there was a write-up mentioning about both of our paper, that this is the way the human must have migrated. So it holds true even today. After that, when the human genome was sequenced, there are 1 million genetic markers are now available for one experiment. Therefore, we used the 1 million SNPs from the nuclear genome. By now, at that time in 2009, we have analyzed about 20,000 individuals, and we carefully selected individuals belonging to all the linguistic group, Dravidians, Indo-European, Austroasiatic, Tibeto-Burman, and Andamanis. We also carefully used the social structures, the so-called upper caste, middle caste, lower caste, tribes, and primitive tribes, to have a representative population from all the groups. And we used the human genome diversity panel and also HapMap data set which was available. And we came to a conclusion about how the human, particularly in India, has been arrived. Okay. So this is again the first evidence suggesting that we in fact reconstructed the Indian population history, where we have shown that these are all Indian populations. This is a European, African, and Chinese, where you see that there are many individuals in the tight cluster, suggesting that genetically they are more homogeneous, not much difference. But look at Indians. Every single population is very unique, right? They are very unique. Again, they are not clustering here and there, suggesting that each population is unique, but still we have some kind of genetic affinities. Okay. And to give you further that how we have classified that the Andamanis are the first one to migrate out of Africa, the deep-rooted divergence. While Andamanis were migrating, some groups stayed in southern part of India, whom we call as ancestral South Indians. And there was second wave of migration, brought people towards north. One group has gone to Europe, and other group has come to northern part of India, whom we call as ancestral North India. These are two founding populations of Indian subcontinent. And these two have admixed, which I will tell you in coming slides, ANI and ASI. And what we conclude here is that all Indians have admixed. That's one of the important points what I wanted to highlight. And we wanted to refute the Aryan invasion theory, and we also predicted there could be large recessive disease in Indian subcontinent. So in addition to these two founding groups, we know that there are large number of population who have migrated recently, including Siddhis, Jews, and Parsis. To give you how they are today genetically, if you take Siddhis, who have been brought by the Portuguese traders from Africa about 400 years back, this is autosomal mitochondrial DNA and Y chromosome DNA, which inherit from father and the mother. This is African genome, Indian genome. You can see how the Siddhis genome is at present is a combination of both. And look at the Jewish. Again, they have come during 5th and 10th century, and this is called structure analysis using 1 million marker. I will not again go into detail. The color indicate, you can correlate that with the genetic component. If you see India and Pakistan have almost similar kind of genetic component. Southeast Asia has different, right? If you look at this, Jewish population, they are somewhere here and here, and they have very tiny proportion, here, they have tiny proportion of Middle East component, and majority of them are mixed with local Indian. That's why they have majority of the Indian genome. In Parsis, again you know that they are the population who maintain highly endogamy, and this is the Parsis genome, and they still hold majority of their original genome, but still there are a lot of admixture. Then putting all of this together, I told you there are two founding populations, there are many populations who have migrated, a few very highlighted, otherwise there are many more such populations. So all of them put together makes India as one of the very complex nation in terms of the genetics. And we thought that analyzing their genome would reveal a lot more information. And right now, if you see how many populations are there in India, there are 4,635 anthropologically well-defined population groups. And the 532 are tribes, 72 are primitive tribes, 36 are still following hunters and gatherers lifestyle. And again, coming back to the question about the ancestral South Indian and ancestral North Indian admixture, to understand that we took another study, taking a lot of sample from south and a lot of sample from the north, based on the recombination, because every generation, when the chromosome is passed on to the next generation, there will be recombination between the homologous chromosomes. Taking that into account, we try to understand when this admixture took place. So in this study, what we have shown, initially ancestral South Indian and ancestral North Indians are the two groups, and they gave rise to many populations initially. Maybe you can imagine the first settlers settled in jungle. After finishing food, some group migrated to other place. Similarly, it might have happened many times. Then what happened during the last 2,000 to 3,000 years, there was admixture. The color indicates that there is a mixture of the genome. Then after admixture, last 2,000 years, every single population started maintaining the endogamy. Endogamy means marrying within the group. Okay? So initially everybody was these two groups unique, then they admixed, then everybody is following the endogamy. Okay? That's the reason that in the previous slide where I showed the PCA, principal component analysis, every group is unique, but still there is a kind of genetic affinities between the group. But what I am going to take you through now quickly, I have very short time, so how the endogamy, marrying within the group, has impact on our health and disease. To understand that, we took another study where we wanted to go beyond India, because the reason is that in one of our study, we have shown that the South Asians, majority of them, have almost similar genetic component, and India in fact gave about 50% of the genetic component to the next, our neighboring countries. Therefore, we thought whatever we say about India is also true for other South Asian countries. This is one example. The second example, in terms of disease-wise, we identified a mutation in a gene called myosin binding protein C3, which causes a disease called cardiomyopathy. And the mutation is in one of the pairs, it's called as heterozygous. It causes disease after 40 or 45 years. But if it is in the homozygous condition, that means both the copy of the chromosome carry the mutation, which leads to early cardiac death, that we confirmed by looking at the post-mortem tissue of the children who died due to cardiac arrest. So we wanted to see how this distributed all over India. We found that it is distributed throughout India about 4.5%. When we have gone beyond that to look at its distribution all over the world, to our surprise we found that is restricted to only in South Asian countries. And probably you can imagine that like that there are many mutations we found, and that could be one of the reasons why the South Asians or Indians are more prone for cardiovascular diseases. Coming back to the study which we wanted to undertake, and these are the places where we sampled, taking representative groups from all the linguistic groups and caste groups, and we come up with another output saying that there's a lot more to study in the South Asian groups, and there's a lot more population-specific recessive disease exist in Indian subcontinent or South Asian population. So this is another study which actually gave a lot more information. What we have shown is that, again, I'm not going to technically, that this is called as IBD score, identity by descent, in simple way, that if you take two unrelated individuals, and if you look at their DNA, if they are really unrelated individuals, you will find more variations. And if they are related several generations back, you may not find any variation between these two sequences. Based on that is calculated. The number of the length of this indicates how many fold the IBD. The more IBD, there's a more chance of having population-specific mutations, may lead to population-specific diseases. Example is that Ashkenazi Jewish and Finnish population, they are with very tiny IBD score, but still they have a lot more diseases. You can see this is in Ashkenazi Jewish, so many diseases, most of them are autosomal recessive. This is a Finnish population, again, lot more diseases. To give you in a simple way that how does it matter and how it is inherited. If the disease causing mutation is dominant, that means one chromosome is causing the, having the mutation in a heterozygous condition, it will express the disease. But the problem of recessive is that one chromosome is normal, one is affected, the individual will not show any disease. It will be a carrier. Most problem exists there. This is the pedigree. Those who are not familiar, when you put circle, that means female, square male, and if you put line, that means these two are married, and these two are their offspring. So it goes like that. And predict that there's a mutation here, It's a recessive mutation, therefore there's no disease. It is passing on to the next generation, again there is no disease because it's going in heterozygous condition, generation after the generation. It's going generation after the generation. So now these two might have lost their relations. Try to trace if they are related or not. Time up, maybe quickly. Can I take a few more minutes? So now these two individuals believe that they are unrelated, but from the same community, right? And if there's a possibility of marrying, and both will contribute this abnormal chromosome to the next generation, therefore there's a disease. I'll tell you one example from our previous study. You can see this is the wild type, this is C, this is a full mutant, both the chromosome carrying the mutation. This is a heterozygous. One is like the wild type C, other chromosome is having the mutation, that's the T. Both the parents are heterozygous, that's absolutely fine. But you can see, unfortunately, both of them transmitted the T, which is having a mutation, and the two children have the disease, and unfortunately, they die at a very early age. So these are the informations which are not actually known even to our medical community. There are many populations which are in village are suffering this kind of diseases. So quickly, I wanted to tell you that we need to follow something like what the Ashkenazi Jewish follows. They have an organization called Dor Eshim. It's a community-based organization. This organization goes to school, collect the sample from the children, sequence, and put it in the database. When the children grow and they are ready to marry and they go to the organization match the DNA of both the individual and see whether they carry the mutation which causes the disease. Both of them carry, then they give advice accordingly. And to take it further, just 2 more slides, we wanted to analyze, although we don't have any clue about the disease aspect of all these populations, but we selected a few populations like color, okay, which has a high IBD score. We also took the some more population which are not mentioned here, but they are in the line. One example I wanted to show is the color population, which has a high IBD score. And we found there's a disease called junctional Hurlitz epidermolysis bullosa. This is a skin disease with congenital heart disease. We sequenced the exome and found there's a 11 base pair deletion in the LAMB gene which causes this. And parents are heterozygous children. So there are 8 such families we identified, and we are going to screen the entire population and identify appropriate counseling will avoid such kind of children. So towards that, there are many more such examples, anti-kidney disease, Udanam kidney disease, and now we have a project funded by Department of Biotechnology to look for such rare diseases, and where we have 20 different centers across India who are actively engaged in genetic analysis. We do the whole genome or exome sequencing to understand whether the mutation is really cause for the disease. We create animal model, and we also try to treat some of the disease using CRISPR-Cas9 technology. And we actually launched very recently, and it was inaugurated, the lab was inaugurated by the former Vice President, Sri Venkaiah Ji, in CDFD, and through which we try to understand some of the rare diseases and try to help the population. So with that, sorry that I took a little more time. I'd like to thank all my colleagues, collaborators, and the list is very long, and also the funding agencies. And thank you very much.